everyone. Welcome to the lectures of BMY4302 Virology. This lecture is brought to you by the Department of Microbiology, Faculty of Biotechnology and Biomolecular Sciences, University Putra Malaysia. I'm Dr. Eddie Chia, and the topic that I would like to share with you today is virus classifications. So, I'm sure all of us have seen this taxonomy of life, where the simplest unit of the taxonomy will be the species. Going up, we have genus, family, order, class, phylum, kingdom, domain, and eventually is life. So when I was a student, when we learned the taxonomy of virus, the highest taxonomic unit that we learn is the order, which is here. So this is one of the examples of the virus taxonomy. The highest is Mononega viralis, then with family Paramyxoviridae, subfamily Paramyxoviridae, and then we have genus, and of course we also have the species. Well, this is quite an old taxonomy for virus. In 2018, the taxonomy of virus has changed tremendously. Not only that we have orders, it moved to class, subphylum, phylum, and realm. So this is what we learn about science, where the science is continuously progressing, and here we can see that the virus now go all the way to realm, but they are not part of the domain because virus practically is not considered as living organism. Right? So these are some of the examples of viruses of different families. I'm showing here the families of viruses that infected vertebrate. So you have DNA virus, we also have RNA virus, and in DNA virus we have double-stranded DNA, virus of single-stranded. We have RNA, where the RNA also have a double-stranded RNA, single-stranded RNA of positive orientations and negative orientations. Also, we have viruses that infecting the plants, double-stranded, single-stranded, double-stranded, and single-stranded of RNA. Of course, we also have family of viruses that infecting bacteria. Same, we have double-stranded and single-stranded DNA, and double-stranded and single-stranded of RNA. So basically, what we learn from here is viruses can actually infect all living organisms in this earth. Well, one thing of very interesting is recently we found that virus can also infect other viruses. This is practically new, and a lot of research have been working on it, and we are looking forward to see more outcome from the research. Here comes the central dogma of molecular biology. I'm sure all of us know it starts with DNA, then when the DNA want to make more of the DNA, it will undergo replication. So you have DNA to produce more DNA. Alright? And from there, we know that the DNA will be transcribed, undergo a process called transcriptions, transcribed from DNA into mRNA. So we can see the flow of the genetic information from DNA down to RNA. So the mRNA or RNA is the messenger RNA, where it carries the specific genes, which will then be translated into protein. So this is the central dogma that we learn all this while. But viruses, being viruses again, and they just mess up all this central dogma. Not only we have DNA, this is the standard, from DNA to RNA, and then from RNA to protein. This is the standard flow of central dogma. However, we also have the virus that do something differently, where we can see here the RNA is then reverse transcribed. See, transcribed is from DNA to RNA, but the virus reverse transcribed the RNA into DNA. So this is very interesting process, where the enzyme that carry out this process is called reverse transcriptase. So the virus that is fall into this category is the HIV. Of course, we also have RNA. Normally, the RNA will be translated into protein, but now we have RNA that transcribe instead of into the protein. They transcribe it, translate into a protein. The RNA transcribe themselves into a different orientations of the RNA, and this RNA will then be used as a template to transcribe again back to the original orientations of the RNA. So this shows that the viruses has gone through a lot of evolutions, okay, to make themselves to adapt to this molecular biology. So viruses are quite smart. So there are a few terms that I would like you to take notes, especially on the enzymes that involved in this process. Of course, the DNA that produces DNA is called DNA polymerase. But if you want to make it more specific, because the template that they use is DNA, we can also call this DNA polymerase as DNA-dependent DNA polymerase. Again, DNA-dependent DNA polymerase. Alright? So here, just now we mentioned that the enzyme that use the RNA to transcribe itself into DNA is called reverse transcriptase. So if you want to make this as part of the DNA-dependent DNA polymerase, you can also say it is an RNA-dependent DNA polymerase for this. Alright? Of course, now we have the DNA that produce the RNA transcriptions. So we have RNA polymerase or DNA-dependent RNA polymerase that produce the RNA. Alright? So the enzyme that involved in transcribing the RNA to RNA, so the template is RNA, so now it becomes RNA-dependent RNA polymerase. Alright? I hope you are clear with this term. So if the template that used by the enzyme is DNA, we call it DNA-dependent DNA polymerase. If the template is RNA, then it can be RNA-dependent, whether they can be a DNA polymerase or RNA polymerase. Alright? Hope you are clear with this term. Right. So, double helix DNA. So we know that double helix DNA is curled as shown in this diagram. So we have a positive strain and a negative strain. What does the positive strain mean? So double helix, if you look at this purple one, this is the, let's say it is a positive strain. The strain that complement to it, it must be at a different orientations. That's why the blue color, if the red purple color is positive strain, then the blue color will be the negative strain. Okay? So this positive strain and negative strain DNA is very important for the transcriptions. For example, in here, what we have here is mRNA, the process of DNA to RNA. So this process is transcriptions. So in order for these transcriptions to happen, it requires the template from the DNA to synthesize this mRNA. So what is the strain that is being used in this case to synthesize this? And we can see, in order for you to synthesize something that complement, it must be of a different orientations. So in this case, the DNA that being used that serves as template for the RNA synthesis will be the negative strain of the RNA. So this negative strain of the, negative strain DNA. This negative strain DNA will be used as a template to synthesize the positive strain mRNA. Alright? So I want you to remember that all the mRNA that being used to synthesize a protein or being used for the process of translation into a protein is a positive strain. So what does a positive strain mean? I hope you remember how does a nucleotide pictures look like here. This is a nucleotide molecule. So we have the ribose, and we have the base, and we also have phosphate group. These are the three major groups of a nucleotide. So whenever you have part that is positive strain or 5', the phosphate will always be the 5', whereas the OH here will always be the 3'. Alright? So when we say that is a positive strain, means that the beginning here is always the 5', the end here is the 3'. Remember this, positive strain, 5' to 3'. What about negative strain? If it is a negative strain, then you have 3' on the left and 5' on the right. Alright, I hope you are clear. Positive strain, 5' to 3', negative strain, 3' to 5'. Alright, so the mRNA that being used to synthesize a protein will always be a positive strain or a 5' to 3'. Alright, so now that you understand what does the 5' to 3' of positive strain or negative strain means, so what we can see here, the green color, viral mRNA is 5' to 3'. Okay, 5' to 3'. So if a virus is positive strain RNA, plus strain RNA, or positive strain RNA, the orientations of the nucleotide will be 5' to 3'. Okay, 5' to 3'. If it is a negative strain, then the RNA will be 3' to 5'. 3' to 5'. Plus strain DNA, 5' to 3'. Negative strain DNA, 3' to 5'. So the difference between the DNA and the RNA, I hope you should know by now, is that in RNA, we will not have T. The T, thymine, will be replaced by uridine. Okay, there is no T. The T will be replaced by U in RNA. So it's very easy for you to recognize if you look at the sequence and the sequence contains U, that is an RNA. If it contains a T, that is the DNA. So now we come to the classifications of the virus by using Baltimore system. Okay, this Baltimore system describes that they classify the virus based on the way the virus replicates and the way the virus translates the protein. Okay, so we have seen this one. So here they have 5' phosphate group. Then the 5' phosphate group always attach itself to the OH group here. And then you get more and more and more. Alright, so this is 5'. Over here, if you have OH group that is free, this will be the 3'. So 5' here and 3' here. Alright, so let's look at the first group of the Baltimore system. So Baltimore system classified the first group of virus based on the double-stranded DNA genome. When the virus has double-stranded DNA genome, the genome replications is double-stranded DNA. Then they will produce another double-stranded DNA. Because the virus wanted to replicate, so they need to produce the same orientation, the same condition of their original genome. So double-stranded DNA will come back to double-stranded DNA. Quite straightforward. Use negative strain to synthesize the positive strain. Use the positive strain to synthesize the negative strain. So in the end, you will have two double-stranded DNA. What about the mRNA synthesis? So the double-stranded DNA, like I mentioned to you, mRNA is positive strain. So what they do is they use the negative strain as a template to synthesize the mRNA. So here we can see that if the virus is double-stranded DNA virus, they already fall into group 1 of Baltimore system. So let's look at second group. Single-stranded DNA genome. So we have single-stranded DNA genome, and we know that if the virus single-stranded DNA is negative strain, so they will use this negative strain to synthesize a positive strain. So it will now become a double-stranded and then use this positive strain to synthesize their negative strain DNA. Right? I know it's a bit confusing. Let me repeat again. So this single-stranded DNA, let's say it is a negative strain. Okay? So the orientations of the genome is 3' to 5'. So in order for them to produce more of the 3' to 5', first what they need to do is the DNA polymerase need to synthesize the single-stranded DNA into double-stranded. So now we have double-stranded DNA. One is from 5' to 3', the other one is from 3' to 5'. So now that we have double-stranded DNA, in order for it to produce more of the single-stranded DNA, which is negative strain, they will use the newly synthesized positive strain DNA to synthesize the single-stranded DNA strain. Alright, this is how the single-stranded DNA genome is being synthesized. Alright, so next, the mRNA single-stranded produce double-stranded DNA, then they will use the negative strain to synthesize the mRNA, which is a positive strain, okay, which is 5' to 3'. So if the virus is single-stranded DNA, it will fall into group 2. Now go to group 3, double-stranded RNA genome. How they replicate the double-stranded RNA will be used to synthesize a single-stranded RNA, and this single-stranded RNA will serve as a template and then produce more of the double-stranded RNA. Okay, how does the mRNA is being synthesized? So double-stranded RNA have both 5' to 3' and 3' to 5'. So this double-stranded RNA can immediately be used as an mRNA because the 3' to 5' is the negative strain and 5' to 3' will use as an mRNA because mRNA is 5' to 3'. So the 5' to 3' of the double-stranded RNA will be used directly as the mRNA. So next is class strain RNA genome or positive strain RNA genome. If a virus is a positive strain RNA has a positive strain RNA genome, so the replication is the RNA will be used, positive strain RNA, to synthesize a negative strain RNA and use this negative strain RNA as a template to synthesize more of the positive strain RNA. What about the mRNA synthesis? The mRNA, positive strain mRNA, positive strain RNA of the genome can be used directly as a mRNA. Alright, so you can see the sign is slightly different. So this double-stranded RNA, the negative strain RNA can be used as a template to synthesize positive strain mRNA. Right? So I might say something wrong just now. The double-stranded RNA, where the negative strain will serve as a template to synthesize the mRNA, which is the positive strain RNA. Next, we go to group 5, negative strain RNA. Negative strain RNA synthesize a positive strain RNA that serves as a template for them to synthesize more negative strain RNA. What about the mRNA? So we know that mRNA is positive strain, so we can't use the negative strain RNA as a template for translation. So this negative strain RNA has to be synthesized into mRNA so that this mRNA will then be used for translation. Then in group 6, we have single-stranded RNA genome. Well, this is the HIV group that is slightly special when it comes to their replications. Single-stranded RNA, where this single-stranded RNA, instead of forming themselves into double-stranded RNA, it will reverse transcribe themselves into double-strand DNA. Alright? And this double-stranded DNA will then be used to synthesize single-strand RNA for their viral genome. So this is quite similar to transcriptions, right? Okay, so mRNA synthesis, single-stranded RNA will then reverse transcribe into double-stranded DNA, and this will serve as a template to synthesize mRNA, similar to here. Alright, so now we have the last group of double-stranded gap DNA. So this particular group of virus is very special, especially one that is well known to us is hepatitis B virus. So the genome of the hepatitis B is gap double-stranded DNA, meaning that the double-stranded DNA is not complete, where one of the strain has synthesized no nucleotide, nucleotide, no nucleotide, nucleotide, and no nucleotide. So that's why we call it a gap double-stranded DNA. So now, when this virus injected their genome into the host, the gap double-stranded DNA will be repaired to produce a double-stranded DNA, which is a complete one. And then this complete double-stranded DNA will be used to synthesize RNA, and this RNA will then reverse transcribe into DNA. So unlike HIV, where reverse transcription happened at the very beginning, but hepatitis B, the reverse transcriptions happen after the transcriptions, and this transcription produce mRNA, and this mRNA will then be reverse transcribed into DNA, and this DNA will continue to produce a gap double-stranded DNA. We'll learn this particular virus later in maybe 2 weeks or 3 weeks later, specifically focused on the hepatitis B virus. What about the mRNA synthesis? So the double-stranded gap double-stranded DNA will complete themselves, become double-stranded DNA, and then this double-stranded DNA will be used to transcribe into mRNA. So based on this system, we can basically classify the virus into 7 groups. Group 1 to group 7. So now, before we proceed to individual group of the virus, which we'll study in detail, and I would like you to understand more about the protein translation process, because this is very important for you to understand the following chapters or following lectures, especially when it comes to virus infections. How does the virus produce their RNA or DNA, and how does this DNA or RNA is being transcribed translate into a protein. So this is a little bit of recap of what protein translations mean. So I'm sure all of you remember when it comes to translations, mRNA will be bound by ribosome, and this ribosome will move along the mRNA and then start synthesize the amino acid. So I also hope you remember there is 3 sites, EPA, acid, peptide, and then attach. So what does it mean? That let's look at these components. When it comes to translations, we will have mRNA, which 5' to 3'. See, remember, positive strain of RNA. mRNA is a positive strain, so the 5' to 3'. Alright? So we have the ribosome bound to the ribosome bound to the mRNA, and then this ribosome will continue to move, and then we have tRNA that contain anticodon that complement to this mRNA that carry an amino acid. So what will happen here is the tRNA bring the amino acid. First thing what they will do is they go into the A site. If they are complement, they will complement themselves to the mRNA, and then as the ribosome move, this peptide bond will be synthesized. So imagine this ribosome is moving, so tRNA will be out, then this will move to here. Alright? So now lysine went in, acid moved to the P site. When lysine moved to the P site, peptide bond will be synthesized. And next, here you have UGC. UGC is complemented by ACG, which is cysteine, and now cysteine will go into the A site. So this process will be continued until they see a stop codon. So this is something that all of us know when you study protein translations. However, what I would like to go further is the detailed mechanism of these translations. So this is very important because we can see how the virus overcome this thing in order to make the cell to produce their viral protein. So from here, I also make another video, a video that I made a couple of years ago for the student to understand more, because this is a mechanism that is quite complicated, and by having that video, the student will be able to keep on watching until they fully understood the process. But briefly, what I will go through with you here is to show you how does the DNA, genetic material, DNA, be transcribed into mRNA, and how does the mRNA to be processed in the nucleus before it is a fully matured mRNA. So over here, you have DNA, 5' to 3', 3' to 5'. Okay, so you will have the 3' to 5' DNA to be used to transcribe the mRNA. So now we have mRNA, and we also know that in your DNA, separate the gene into a few several fractions. They are not for example, one particular protein. Maybe this protein, the genes that coded for this protein is not always together. They are being separated into a few fragments. All right. So, and these few fragments is what we call the exon, where the gene is separated by intron, which is the light beige color here. So, in order for you to have a proper protein, you have to remove this intron. Right. So all these things happen in the nucleus. So you have double-stranded DNA, then mRNA being produced through transcriptions. So now we have an immature mRNA that contains exon, intron, and so on. So this mRNA, premature mRNA, need to be processed before it can be used as a mature mRNA for the translations. Three things. Please remember these three things for a mature mRNA. First thing is it needs a cap. The 5' of the mRNA is always capped. Alright? 5' of a mature mRNA is always capped. So this cap contains of methyl 7-guanine, okay, M7G. So this M7G will bind themselves into with the first nucleotide of the mRNA. Right? This is the first characteristic. The second characteristic is it must have a poly-A tail. Right? Poly-A tail, poly, many, A. Okay? So, and you can see there's a tail here, means that this is located at the end. So, mRNA start mRNA with cap and with poly-A tail. You have A up to hundreds of A's of an mRNA. So these are the second characteristic. First characteristic, cap. Second characteristic, poly-A tail. Now, third characteristic, it must go through splicing. So it must go through splicing to remove all these intron so that the exon can be bound together to become a matured mRNA containing only the sequence that is important to produce that particular protein. And now we have a complete or a matured mRNA for the translation process. First, you have cap. Second, you have poly-A tail. and lastly splicing to remove all the intron, then you have a complete mRNA. So an mRNA has to be complete in this form before they can move out from the nucleus into the cytoplasm. So translation happen in cytoplasm. Transcription happen in nucleus. Alright? So when this is ready, then it will move out from the nucleus into the cytoplasm for the translations to happen. So as I shown you just now of this diagram, this is what we learned. Alright, so you need a ribosome that binds to the mRNA to produce the amino acid or the peptide or even the protein. Before that can happen, it actually goes through a very complicated process as shown in this diagram. So I would like you to watch the next video for a complete understanding of this process. So I'm not going to discuss about this now. Please watch the next video. Alright, so this is also part of the translations, and I hope you can understand that. So I also give you the detail of explanations and descriptions of how the process is being done, but I would like you to watch the video and try to write your own script, because when you learn how to write, it will help you when it comes to describing it, not only for your own understanding, perhaps, okay, it might come out in the exam, who knows. Alright, so once you understand this, you will understand a lot more about viral protein translations in the host cell. So after you have watched this video and the next video, what I would like you to do is go back to Putrablast, okay, look for this PDF file. Okay, one is Picorna Viride, poliovirus replication and poliovirus. These are the PDF file that I've uploaded in your Putrablast. I would like you to either, if you like it printed, print it. If you prefer to read it online, please read it before you watch the next video on poliovirus, which I will upload maybe a few days later for Thursday class. Alright, that's it. So if you have any questions, please let me know. I'll create a forum so that we can have a discussion in the forum. Thank you.